I first got interested in birds when a neighbor in Riverdale, New York, took me bird watching when I was 8. And that neighbor has probably long since stopped bird watching, and I just haven't turned back. My lab is interested in the evolutionary history and the evolutionary biology of birds. These are house finches. They're a common North American songbird, and we've been studying the interaction of this species with a pathogen, a disease. It's a type of bacterium called mycoplasma. We're also studying the evolution of the pathogen itself. And what we're interested in is how the bird has evolved in response to this very powerful selective agent. Disease is one of the most powerful agents of natural selection that we know of. Our colleagues and I have documented changes in the way the birds look and their overall shape and size and color in response to this disease. For example, it turned out that birds that were more red were better survivors. The yellow birds and the drabber birds tended not to survive. And for that reason, we're very interested in the possibility that the red plumage of the house finch might be a signal for superior genes, genes which might give it resistance to this pathogen. And this sort of research has a lot of implications for how humans might respond to disease. We know a lot about this particular interaction, when the pathogen arose, how quickly it spread in the house finches across the eastern U.S. And so it provides a nice system for understanding in detail the dynamics of a disease that's newly emerged in a new host. And we need those sorts of models so that we can compare similar situations in humans. Australian birds are fascinating and unique because they have diversified in a variety of different habitats completely in isolation from the rest of the world. And that makes them a really wonderful evolutionary experiment to study. We've got a couple different songbirds in the net here. These are all species endemic to Australia, not found anywhere else. We are using genetic techniques to study the evolution and speciation of Australian birds. And we're interested in how species on the eastern side of the continent have diverged from species on the west. Here's a black-tailed tree creeper on the west and a brown tree creeper on the east. These are two related species, but they're found in different parts of the continent. We also have, for example, long-tailed finches, such as this individual here. Here's a long-tailed finch. You can see its long, pointy tail. And a black-throated finch, which you can see right here. You can see these two species are very similar to each other, but they've speciated. One has a long, pointy tail. The other one has a stubby tail. One has a darker breast. The other one has a lighter breast. And we're using genetic techniques to study when these pairs diverged and whether the different pairs diverged at the same times or at different times. Just a tiny drop of blood is enough to fuel hundreds of PCR reactions. We'll spread its wing here. By linking the time points given by the DNA sequences, this can tell us a lot about the past environments that they speciated in and whether environmental change caused a whole suite of birds to diversify at the same time. All right, she's ready to go. There we go. Come on, come on, come on. There we go. There he goes. He's okay.